Hello everyone, welcome to Tawhid Foundation at Allied Healthcare Sciences, Bangalore. Today's class is about Parasitology Part 3 for BSc MLT second year students. Coming to introduction part, what is parasite? A parasite is described as an organism that derives sustenance from another living organisms, the host, without reciprocating any benefit. The parasite relies on the host for nutrients and survival, often affecting the host health and biological functions. Second is, what is the purpose of laboratory diagnosis? So, importance of laboratory diagnosis is highlighted as a crucial process for detecting and confirming parasite infections. Accurate lab-based identification is necessary not only to verify clinical suspicions but also to reveal infections that might be unnoticed in the absence of symptoms. So, timely diagnosis guides effective treatment, helps prevent the spread of disease, and informs public health strategies. Third is methods for laboratory diagnosis of parasitic infections. So, here the main techniques employed for diagnosing parasitic infections first is microscopy. This involves direct visualization of parasites in biological specimens using microscopes. It remains the cornerstone of diagnostic parasitology. Second is culture. So here growth of parasites is in specialized laboratory media for identification and further studies will be used. Third is molecular techniques such as PCR, which allow highly specific detection of parasitic DNA or RNA. So employing these three methods, which is microscopy, culture, and molecular techniques, ensures a comprehensive approach to identifying various parasitic organisms. So these three method is included. Last is materials required and procedures. So this slide also previews the typical materials which require, example glass slides, cover slips, reagents, staining solution, microscopes, and the stepwise procedures for specimen collection and analysis. So understanding these elements equips students with both the theoretical knowledge and hands-on skills necessary for successful laboratory practices in parasitology. Next is objectives. So objectives of laboratory diagnosis. So this slide emphasizes the broader impact of laboratory diagnosis in parasitology. It informs population level surveillance, helping public health authorities track and analyze trends in parasitic prevalence, distribution, and drug resistance. Early identification and accurate diagnosis enable targeted intervention programs, ultimately helping reduce diseases burden in affected communities and improving public health outcomes. So these two is the objective. Laboratory diagnosis contributes to the surveillance and monitoring of parasitic disease at the population level. Second, by detecting cases and identifying trends in parasite prevalence, distribution, and drug resistance, public health authorities can implement targeted interventions and control strategies to reduce disease burden. Next is, what is parasite? All these terms definition is very important in this parasitology part. These three terms, parasite, host, and vector. So parasite again is an organism deriving sustenance from another without making compensation. The uncompensated organism is the host, which means an organism living on or inside a host deriving nutrition without offering any benefit in return is nothing but parasite. Second is host. So refers to an organism that carries another organism, typically a parasite, providing it with a place to live and often serving as a source of nutrients, which means the organism that supports the parasite, providing it nourishment and habitat. Third is vector, is the carrier of a parasite from one host to another, often an insect. So commonly an insect, a vector is responsible for understanding transmitting parasites from one host to another. So these three concepts are fundamental in parasitology for understanding transmission cycles, pathogenesis and epidemiology. Next is classification of parasites. So this slide covers the systemic organization of parasites into three major groups, which is protozoa, helminthes and ectoparasites. So protozoa are single-celled organisms that cause disease such as malaria, amoebiasis, giardiasis and trichomoniasis, often transmitted via contaminated water or vectors. Helminthes are multicellular worms divided into nematodes, that is nothing but roundworms, cystos, that is tapeworms, and trematode, which is flukes. So with complex life cycles involving host and often intermediate host. Ectoparasite include all external parasite like lice, fleas, ticks, and mites, which can directly cause disease or act as vectors for other pathogens. So, understanding these groups, their life cycles, routes of transmission, and diseases they cause informs how medical laboratories select diagnostic tests and clinicians prescribe treatments. Diversity among protozoa. So, the diversity among protozoa, this slide visually represents the variety of protozoan parasites, emphasizing differences in morphology, structure, and life cycle stages. So, I'll give you a detailed explanation of each of the four images typically found in this slide showing protozoan diversity. So, first is nothing but amoeboid protozoa. Example is Antiamoeba histolytica. So this image depicts an amoeboid protozoan characterized by its irregular shape and pseudopodia, that is temporary projections of the cytoplasm. Amoeba move and feed by extending these pseudopods, engulfing food particles or host cells. In pathogenic species like Antamoeba histolytica, this mobility enables tissue invasion, causing amoebic dysentery. So this image shows both trophozoid, that is active feeding stage, and cyst, which is dormant infective stage, forms important because cysts survive in the environment and transmit infection. Second is flagellated protozoa. With example is Giardia lamblia. So this image shows a flagellated protozoan such as Giardia lamblia with distinctive paired nuclei and multiple flagella. So flagella are whip-like appendages used for locomotion, allowing the organism to move through liquid environments such as the intestinal lumen. Giardia causes giardiasis, a common cause of diarrheal illness. So this image highlights the trophozoite stage which colonizes the small intestine and also shows cysts responsible for transmission via contaminated water. Third is ciliate protozoa. This image illustrates ciliated protozoa which possesses numerous short hair-like structures which is cilia covering their surfaces. Cilia beat rhythmically for locomotion and to sweep food particles towards the cell's oral groove. While many ciliated protozoa are free living, some may be opportunistic parasites or commensals in humans. So this picture demonstrates the complex surface morphology and ciliary organization that distinguish ciliates from other protozoa. Last is sporozoan protozoa, which is Plasmodium species. This final image represents sporozoan protozoa exemplified by Plasmodium species, the causative agents of malaria. These protozoa are non-motile and have complex life cycles including sporozoites, merozoites, and gametocytes. Stages often seen in blood smears. The image might show infected red blood cells or different parasites, so forms illustrating their intracellular parasitism. Sporozoans reproduce sexually and asexually, typically requiring both vertebrate and insect host for life cycle completion. So together these four images highlights the morphological and functional diversity among protozoa, amoeboid movement, flagellar movement, motility, ciliary locomotion, and sporozoan intracellular parasitism. Each important for understanding their biology, pathology, diagnostic features, and control measures in parasitic diseases. So, the diagnosis of parasite diseases rests on two fundamental pillars, which is clinical evaluation and laboratory testing. Both components complement each other and are indispensable for accurate detection, management, and control of parasitic infection. Diagnosis of parasitic infection. So, diagnosis is done through clinical evaluation, laboratory testing. Why? What is the purpose of laboratory diagnosis? Is for confirmation of clinical suspicion, and second is identification of unsuspected infection. We'll start with clinical evaluation. Clinical evaluation involves a thorough assessment of the patient's syndrome, symptoms like medical history and potential exposure to parasites. Symptoms may vary widely depending on the type of parasite and the organs affected, ranging from gastrointestinal disturbances like diarrhea and abdominal pain to systemic signs such as fever, anemia, or skin lesions. The clinician also considers epidemiological factors such as travel history to endemic regions, occupational exposures like farming or working with animals, sanitation condition, and contact with vectors, example mosquitoes, flies. So identifying risk factors and symptom patterns help generate a strong clinical suspicion and guides appropriate laboratory investigations. However, many parasitic infections can remain asymptomatic or produce non-specific symptoms, which makes clinical evaluation alone insufficient for definitive diagnosis. Second is laboratory testing. Laboratory testing is essential to confirm or rule out parasitic infections suggested by clinical evaluation and to identify unsuspected cases that may not manifest clear symptoms. It offers objective scientific evidence through direct or indirect parasite detection method. Common diagnostic approaches here include microscopy, molecular technique, and serological testing. So, microscopy, the traditional and most widely used method, microscopy involves examining biological specimens, which include stool, blood, urine, tissue, under a microscope to identify parasite forms such as cysts, eggs, trophozoites, or larvae. Staining techniques, example Giemsa stain or iodine, and concentration method enhance parasite visibility. Blood smear, vital for diagnosing malaria or filariasis, reveal intracellular and extracellular parasites. For molecular method techniques, advanced molecular diagnostic like polymerase chain reaction (PCR) detect parasites, DNA or RNA with high sensitivity and specificity. These methods can identify parasites even in low density infections, differentiate species or strains, and help monitor drug resistance. Molecular tests are particularly valuable for infections like malaria, toxoplasmosis, amoebiasis, and leishmaniasis. Next is serological test for laboratory testing. Serological tests, these detect host antibodies or parasite antigens in blood or serum, providing indirect evidence of infection. They are useful for chronic infections or when parasites are difficult to observe microscopically. Together, these three tests include microscopy, molecular techniques, and serology testing. So, these three tests provide definitive confirmation, enabling precise epidemiological data collection and informing treatment strategies tailored to the identified parasite, species, and infection stages. Significance of laboratory diagnosis. Why? What is the purpose of laboratory diagnosis? So, laboratory confirmation is critical for reliable diagnosis, especially in cases where clinical signs are ambiguous or absent. It enables early detection of parasitic infections to prevent complications, guide targeted treatment, and reduce transmission. Furthermore, recording laboratory confirmed cases contributes to surveillance efforts, helping public health authorities track disease pattern, emergence of drug resistance, and effectiveness of control program. Without lab testing, misdiagnosis could lead to inappropriate treatment and continued disease spread. So, one in whole, while clinical evaluation forms the initial basis for suspecting parasitic infections, laboratory testing is indispensable for confirming diagnosis, guiding clinical management, and supporting public health interventions against parasitic diseases. Types of specimens. A variety of specimen types are utilized in diagnosing parasitic infections depending on the suspected pathogen and site of infection. Stool for intestinal parasite, which is worms and protozoa. Blood for bloodborne organisms, which is malaria and filariasis. Serum and plasma for indirect testing, which is antigen antibody detection. Other specimens include anal swabs, duodenal aspirate, sputum, urine, urogenital specimens, tissue aspirates collected where specific parasites may localize. Correct specimen collection is essential for accurate laboratory diagnosis. The main infection methods include diagnosis in the laboratory relies primarily on microscopy and culture. Microscopy visual identification of parasites is in stained or unstained samples either directly or following concentration procedures to increase detection of low level infections. Cultures growing certain parasites under controlled conditions for identification, characterization, and drug susceptibility testing, particularly useful for some protozoa or helminthes with specific growth requirements. Both these methods require skilled technique, appropriate materials, and understanding of parasite biology. Stool examination practices. Stool samples are examined under a microscope to detect the eggs, cysts, or oocytes of intestinal parasites such as helminthes and protozoa. Concentration techniques, example sedimentation, flotation, and specialized stains, example iodine, trichome, are used to enhance visualization and detection rates. Specifically, especially for specimens containing low parasitic numbers. Next is molecular methods of identification. Modern parasitology incorporates molecular tools such as PCR, polymerase chain reaction, for direct detection of parasite DNA. Further genetic analysis, including RFLP, that is restriction fragment length polymorphism, or DNA sequencing, allows species identification and investigation of genetic diversity, drug resistance, or epidemiological tracking. These techniques greatly increase sensitivity or specificity for detecting malaria, toxoplasmosis, amoebiasis, and leishmaniasis. Blood examination methodologies. Blood smears are essential for diagnosing bloodborne parasitic infections like malaria, plasmodium species, trypanosomiasis, that is trypanosoma species, and filariasis, that is microfilaria. So advanced chains such as GIMSA and antigen detection assays make it possible to detect even low concentrations of parasites. Blood examination also allow quantification of parasitemia and assessment of disease severity. So the laboratory diagnosis of malaria using blood smears is a standardized process that requires careful preparation and examination to ensure accurate detection and quantification of the parasite. So, the stepwise approach involves several critical components and procedural steps. So, required materials for successful malaria diagnosis begins with gathering the necessary materials. Clean glass slides used to prepare blood smear that serve as the specimen for microscopic examination. Clean cover slips, though not always required for blood smears. Cover slips are sometimes used in related blood testing procedure. Alcohol swabs used to disinfect the patient's finger before blood collection to maintain aseptic conditions and prevent infection. Lancet or needle used to puncture the patient's fingertip gently to obtain a drop of blood. Microscope equipped with appropriate magnification, which is 100x to 400x, essential for visualizing malaria parasites and stain smear. Giemsa stain, a specific staining solution that differentially colors the blood cells and parasites, enhancing parasite visibility and morphological differentiation. Preparation of blood smear. So, collecting the blood sample, the patient's fingertip is first cleaned and dried. A small drop of blood is obtained using a sterile lancet or needle, making the thin smear. A clean glass slide is held at a 30 to 45-degree angle to the drop of blood. The drop is allowed to spread along the edge of the slide. Then, using another clean slide held at the same angle, the blood is spread smoothly and evenly across the first slide to form a thin uniform smear. This thin layer ensures the blood cells and parasites are in optimal monolayer for microscopic examination. Third is drying the smear. The smear is air dried completely at room temperature. It is important not to use artificial heat or blow dry, as this can distort the blood cells and parasites, compromising the examination quality. So, these procedure I explained already. Spread of this blood and smear fixation. We'll go through this once. Prepare these materials, all this required material: glass slide, cover slip, alcohol swab, lancet or needles, and staining solution. Prepare the finger. You should clean the fingertip with alcohol swab and puncture with lancet or needle used to prick the patient's fingertip to obtain a small drop of blood. Alternatively, use a finger prick device if available. Preparation of slide: clean glass slide around 30 to 45-degree angle of the patient's finger. Touch the edge of the slide to the drop of blood, allowing it to spread along the edge of the slide. Spread of this blood using another clean slide. You should spread the drop of blood. Pull the second slide smoothly, spreading the blood to create a thin uniform layer. Air dry the smear. It is very important. Allow the blood smear to air dry completely at room temperature. You should not use heat or blow dry on the slide, which will distort the uh, uh, parasites. So we should avoid doing that. Fix the smear. After fixing, you can visualize it. Once the smear is dry fried, staining, use solutions Giemsa stain. For the recommended time is usually 10 to 15 minutes. Rinse and dry. After staining, the slide gently wash with distilled water to remove excess stain. Allow the slide to air dry completely and then examine under microscope. Thick smear. So it is not fixed in methanol. This allows the red blood cell to be hemolyzed and leukocytes, and any malaria parasites present will be the only detectable elements. However, hemolysis, which may lead to distorted plasmodial morphology, making plasmodium species differentiation difficult. Therefore, thick smears are mainly used to detect infection and to estimate parasitemia. This is the visual representation of thick and thin blood smear. Like this, thin film will be formed, and like this, thick film will be formed. Figure 2 represent infected RBCs under microscopes. Advantages and disadvantages. So, blood smear methods for malaria diagnosis are inexpensive and facilitate both quantification and species differentiation. However, diagnostic accuracy heavily depends on the quality of sample preparation, microscope, slide reading skills, parasite density, and the time spent examining the smear. These methods are not suitable for large scale epidemiological studies but remain fundamental in routine clinical diagnosis. Next topic is arthropods. So, arthropods, entomology and medical entomology. So, this entomology is the scientific study of insects and their relationships with humans, the environments, and the organisms. This discipline contributes to multiple fields such as agriculture, human and animal health, biology, chemistry, molecular sciences, criminology, and forensics. Medical anatomology is a specialized branch focused on arthropods that either cause diseases directly or act as vector transmitting pathogens to humans. Understanding these vectors is essential for disease control and public health interventions. So, entomology is the study of insects and their relationships to humans, the environment, and other organisms. Entomologists make great contribution in these fields of sciences. Medical entomology is the branch of science that deal with arthropods, which causes disease, or that serve as carriers, which is nothing but vectors of organism that cause disease in humans. So basically, what are arthropods? The term arthropod is derived or comes from Greek word meaning joint, meaning which is joint, arthro, and foot, which is pods. So arthropods are small invertebrates characterized by segmented bodies with jointed legs and a hard outer skeleton made of chitin. The flexible membranes between joints allow for mobility. Despite their small size, they are most abundant and diverse life forms adapted morphologically, physiologically, and behaviorally to various environments, making them ecologically significant. So, arthropods have diverse of this morphological, physiological, and behavior adaptations to the surrounding. They are the most abundant form of life. Despite their small size, the huge numbers or biomass of insects that they have significant impact on environment as well as humans. Importance of arthropods. While most arthropods are harmless, some species have great medical importance due to their ability to cause annoyance, discomfort, or disease. They can be classified into four groups, which is harmful arthropods. Second is ectoparasites, mechanical transporters, and vectors. So we'll go one by one. The majority of arthropods are not harmful to humans. However, a number of species are considered medically important because they can cause annoyance, physical discomfort, or disease. Harmful arthropods, it cause nuisance and physical damage such as bites and blood loss. Example is mosquitoes and fleas, bugs, and giants. Second is ectoparasites. It is permanently living on host and feed on blood but do not necessarily transmit pathogen. Example is head lice, scabies, mites, and pubic lice. Third is mechanical transporters. Passively spread disease by contaminating food surfaces with pathogens acquired from feces or waste. Example is flies and cockroaches. Fourth is vectors. It is actively transmitting pathogens where parasites develop or multiply inside them before transmission. Example is mosquitoes transmitting malaria, flies, body lice, and fleas. Why are these arthropods so successful? So, arthropods' evolutionary success stems from multiple factors. Their small size provide access to many ecological niches. High reproductive outputs lead to genetic diversity. Different life stages often exploit varying resources, reducing internal competition. Flight capability enhances dispersal and colonization. Advanced sensory systems aid survival and host detection. Specialized appendages enable adaptations for feeding, movement, and reproduction. So, these points are important for arthropods to be successful. Okay. Arthropod habitats. Arthropod inhabit diverse environments influenced by factors like food availability, breeding media, climate, competition, and natural enemies. Habitats includes soil, water, ambient air, or on humans and animals, on plants. Okay, so depending upon species, arthropods live in various habitats. Factors that control habitats of arthropods include food, disease, breeding media, climate, competition, natural enemies, etc. The soil, it's a surface or subterranean dwellers, that is spiders, scorpions, fleas, cockroaches, etc. It may be found on the surface of the soil or underground. Second is water. Fresh water, salt water, or hot springs inhabitants, for example, crabs, lobsters, crayfish, etc. Ambient air flyers. So, here temporary habitats for flying species include bees, mosquitoes, flies, wasps, etc., though it is not a permanent habitat. Flyers are fast spreaders of contamination and pollutions, therefore, it is hard to control the flyers. Next is on humans and animals. On men, ectoparasites. Ectoparasite forms that feed on blood. These are dangerous groups to health since they feed on human blood. This could be obligatory parasite. Example include loss or intermittent on or off, that is ticks. Last is animals, lice, ticks, mites, fleas, mosquitoes. These are the examples which is causing, including arthropod habitats. Next is on plants, species like beetles and aphids that often affect this plant health. General control methods of arthropods. Control aims to protect human health primarily using methods such as personal protection, environmental manipulation, barrier, biological control, local traditional method, and chemical control. So these are the general control methods of arthropods. We'll go one by one. So control of medically important arthropods employ many principles used against pest or agriculture importance. The basic purpose of controlling arthropod is to preserve health and well-being of humans. On the other hand, control of arthropods affecting livestock, crops, all this fundamentally guided by economic principles. Controlling arthropods including following methods, which is personal protection. Physical barriers between men and arthropod. Example is chemical barriers that repel arthropods from actual biting and arthropod toxicant that are applied directly. Example is given insecticide treated bed nets for the control of malaria and leishmaniasis. Second is environmental manipulation. Breeding site modification like draining marshes to prevent mosquito breeding. Example is drainage of marshy areas for controlling malaria. Third is barrier zones and quarantines. So areas kept vector free require protection to avoid reinvasions. Fourth is biological control, use of natural predators or parasite to suppress vector populations. Fifth is local control methods. Example include using hot ash or aromatic plants to repel insects. Though trial and error, man has established this local way of fighting and controlling arthropods. So last, these following are some of the local controls. as I mentioned, hot ash to kill or drive away insects like ants, hot water against ants, bedbugs, lice, fleas, certain odors, and sticky leaves with white eucalyptus, mimosa, etc. are used as insect repellents. Chemical control: stomach poisons ingested by insects, which is baits, or contact poisons disrupting their nervous system, causing paralysis and death, exemplified by indoor residual spraying as stomach poisons and as contact poisons. Next is modes of disease transmission by arthropods, which is through direct contact, mechanical transmission, and biological transmission. Direct contact transmission through direct transfer of ectoparasites like lice or mites between host. Mechanical transmission: pathogens are passively carried on body parts or via contamination of food by insects such as houseflies. Includes transmission by regulation or defecation. Third is biological transmission. Pathogens multiply or develop within the arthropod vector before transmission. It includes propagative transmission, which is multiplication without morphological change. Example is viruses. Clinical transmission: here developmental changes without multiplication. Example is filariasis. When W. Bancroft or filariasis transmitted in Culex female. Third is cyclopropagative transmission, which is propagative and cylindrical transmission. Here both multiplication and development happen. Example is malaria parasites. Vector capacity. Vectorial capacity measures an arthropod population's ability to transmit a pathogen dependent on abundance. Higher number increases transmission risk. Host preference determines which hosts are targeted, guiding transmission and dynamics. Reproductive capacity, the speed at which vector populations grow. Longevity, the lifespan affects the chance of pathogen transmission. Dispersal, greater movement enhances disease spread. Vector competence, genetic ability to carry and transmit specific pathogens. So these are the things which I explained earlier. Abundance: the more vectors there are, the higher the probability of disease transmission. Host preference and host feeding patterns. Knowing what the vectors feed upon allows for identification of disease transmission. Reproductive capacity, a measure of the rate at which population of vector increases. Longevity is knowing for how long the stages of vector's life cycle will last. Dispersal is knowing how far a vector can fly or move freely. The greater the movement, the greater the chance for spread of diseases. Vector competence, the susceptibility of a group of arthropods to a given strain of pathogens and the ability of those arthropods to transmit the pathogen. This is a genetic characteristics for a particular vector for a given microorganisms. So arthropods of medical importance. Key medically important arthropods include insects. Insects, orders such as diphtheria, true flies, mosquitoes, Hemiptera, which is true bugs, Anoplura, which is lice, and Siphonaptera, which is fleas, and Dicytoptera, which is cockroaches. So these are the insects, 5 groups which are of medical importance. Archan Here arachnids is also there, which include mites and ticks, many of which are vectors, and crustaceans such as cyclops, which play roles in diseases. This so these true flies ordered Diptera, true bugs, Hemiptera, lice, Anoplura, fleas, Siphonaptera, and cockroaches, which is Dictoptera. Crustaceans, again cyclops and crabs. And this arachnids, only mites and ticks are vectors of these diseases. These are the class Insecta. Mosquitoes, anopheles, Culiations, flies, housefly, sandfly, ceases fly, black fly, human lice, head and body lice, crab lice, flea, rat flea, sand flea, and radioid bug. This class include Arcanida, which is ticks, hard ticks, soft tick, mites, Leptotrombidium mite, Tromboculide mite, and itch mite. Classes Crustacea, which is cyclops. So crustaceans. Here, most crustaceans have the following features, which is two pairs of antennae, three pairs of chewing appendages, various numbers of pairs of legs. They differ from insects in that they have legs on the abdomen and the thorax. Medical importance. Cyclops are intermediate hosts of the fish tapeworm and Dracunculus medinensis insect. This image you can refer here, the which everything is given, what derives what. A derives antenna, B which is brain, compound eye, mouth, flight muscle, wing, leg muscle, leg, digestive tract, and reproductive organs, and last is anus. So next is characteristics of insect. Insects have segmented bodies with head, thorax, and abdomen, single pair of antennae, compound eyes, mandibles and maxilla, and exoskeletons with jointed appendages. Mosquito females possesses proboscis to pierce skin and suck blood, crucial for disease transmission like malaria and dengue. Their life cycle includes egg, larva, pupa, and adult stages. So these are the characteristics of insects, how body is divided, what their head bears, single pair of antenna, pair of mandible and maxilla. Visual organs include compound eyes. It may be absent in some flea. Thorax possesses three pairs of legs and usually one or two pairs of wings. Abdominal appendages are modified to terminal genital organs near cucaudal end of the body. Insects of medical importance. So, here, directly cause damage to human tissues. Second is act as vectors for disease causing organism. And last is useful model systems in biological studies. Mosquitoes will go one by one. Here, mosquitoes are insects belonging to the order Diphtheria, which is true flies. Three subfamilies exist. Most important ones are Anopheles and Culinae. Over 3,500 species of mosquitoes have further been described. Example of most common genera are Aedes, Anopheles, Culex, etc. The mosquito life cycle consists of egg, larva, pupa, and adult stages. Females have tube-like mouth part, which is proboscis, that can pierce the skin of a host and feed on blood. Males differ from females by having mouth parts not suitable for piercing skin, which is near nectar is their principal food source. Eggs are laid on the water surface and hatch into motile larvae that feed on aquatic algae and organic material. So this is the example of male anaphile and female anaphile. Male mosquitoes have a bushy antenna and the palpi are as long as proboscis and clubbed at the end. Female mosquitoes have antennae with fewer, shorter hairs. In female anaphiles, the palpi are as long and straight as the proboscis. Mosquito life cycle. So here you can see how they'll complete their life cycle. First, it will grow as eggs, then first larval stage, then second larval stage, then third larval stage, fourth larval stage, and the final pupa stage, and last they'll become adult. So each larval stage is larger than the previous one. This mating occurs between each larval and pupal stage. Larva and pupal stage are aquatic. Mosquitoes are important vectors of parasitic diseases such as malaria and filariasis. Also, arboviral diseases such as it will include symptoms such as yellow fever, chikungunya, West Nile, dengue fever, and Zika. Mosquito saliva is transferred to the host during the bite and they cause an itchy rashes. Anophiles. So these all are the characteristics of insects and mosquitoes. It has this segmented body, single pair of this antenna, compound eyes, mandible maxilla, exoskeleton with joint appendages. Female possesses this proboscis to pierce skin and suck blood, crucial for disease transmission like malaria and dengue. These their life cycle include the main things which is egg, larva, pupa, and adult stage. Next is anophiles. Anophiles and Culex mosquitoes, a genus with hundreds of species, is the primary vector of malaria, preferring breeding in clean water and biting mostly from dusk till dawn. Culex mosquitoes are domestic and active indoors and outdoors as well, transmit lymphatic filariasis and viral infections such as West Nile virus. So about 380 species of anophiles occur around the world. Some 60 species are sufficiently attracted to human to act as vectors of malaria. A number of anophil species are also vectors of filariasis and viral diseases. Female anophile mosquitoes spread the diseases easily. The anophelines that enter houses to feed often rest indoors for few hours after feeding. They may then leave for outdoor sheltered resting sites, among them vegetation, undersides of which cracks and crevices in trees. These many anophil species feed on both humans as well as animals. However, they differ in the degree to which they prefer one over the other. Species of some feed mostly on animals while others mostly entirely on human beings. So anophiles, their mostly preferred breeding sites are pools, slow running streams, certain epiphytic plants, rice fields, and pools of rainwater. Artificial containers such as overhead tanks, cisterns, and pots are not usually suitable. Mosquitoes, anophiles are active between sunset and sunrise. Each species has specific biting hours, and there are also variations in preference for biting indoors and outdoors. How disease is transmitting? So malaria is caused by various species of Plasmodium species, Culex mosquitoes. Culex species is markedly domestic species. However, the adult females bite people and animals throughout the night, indoors and outdoors as well. They are inactive during the day and are often found resting in dark corners of rooms, shelters, and culverts. They also rest outdoors on vegetation and in holes of trees in forested areas. So diseases are transmitted through lymphatic filariasis, tularemia, a bacterial disease caused by Francisella tularensis, arbovirus infection, West Nile virus, and Japanese encephalitis. Vector control strategies. So vector control include individual residual spraying with insecticides, space sprays, protective clothing, insecticide treated bed nets, genetic approaches, sterile male technique, and chemoprophylaxis for travelers. Anti-larval methods involve source reduction of breeding grounds and biological control like mosquito fish. These genetic control, sterile male technique, and gene replacement for travelers visit endemic areas. Chemoprophylaxis provide effective protection and drugs recommended for this. Anti-larval measures. Old anti-larval measures such as oiling the collection of standing water or dusting them with Paris green have now become promising with the increase of insecticide resistance. Source reduction techniques which reduce mosquito breeding size include drainage or filling, deepening or flushing, management of water level, intermittent irrigation are among these classical methods of malaria control. Biological control is use of mosquito fish and Lebestus reticulatus in pets, sewage, and farm fonts. Next is flies and their public health impact. So houseflies proliferate rapidly in unsanitary environments and can mechanically transmit pathogens such as bacteria causing salmonellosis and viruses like poliovirus. So, control programs focus on source reduction, physical exclusion, biological predators, chemical agents, and public educations to reduce breeding and contact. The reproductive capacity, diet, and filth, coupled with affinity for men and animals, make flies reservoirs and disseminators of fly-borne parasites and microbial pathogens. Here, flies proliferate under warm conditions, which will accelerate larval development and maximize adult activity. Disease and public health relationship. Annoyance factors. Here, most humans are intolerant of houseflies hovering around patios, landing on exposed food, and more so when they land on face, arms, and legs of humans. Second is disease produced by microorganism and helminthes. Flies are incriminated in contaminating exposed food with bacteria, which will cause salmonellosis, shigellosis, typhoid, as well as cholera. The poliovirus can also be transmitted via flies. The disease caused by intestinal helminthes or protozoa such as Giardia lambiasis can also be passed on by flies. Management and control. So how you'll control the explosive reproductive potential of these flies and their ability to disperse makes these vectors difficult to control. However, balanced integrated pest management IPM fly program include source reduction, exclusion, biological control, chemical control, public education. So these are the things which will help to manage and control the spreading the diseases. Next is fleas. Blood-sucking ectoparasites primarily on mammals and birds. Fleas can cause irritation, allergic reaction, and serve as vectors for diseases like plague and murine typhus. Controls involves pesticides and limiting rodent host. So, public health importance. Fleas can be a public health concern by being nuisance as well as vectors. Their attack on humans and domestic animals can cause this irritation, loss of blood, and severe discomfort. While some individuals may suffer severe allergic reactions or secondary infections from their bites, flea can vector disease organism which cause bubonic plague, murine typhus, and trypanosomiasis. Fleas also serve as intermediate host for tapeworms and filarial worms. Management and control, as I mentioned, involves pesticides and limiting rodent host. Next is sandfly. Small flying insects with feathery wings and hair bodies, sandflies transmit leishmaniasis and sandfly fever. So these like this look like a mosquito except its body is hairy and the wings are feathery, smaller than mosquitoes. Wings rest over the body like angel's wing. It have large compound eyes and long slender legs. Identification. Male have posterior end of abdomen which carries prominent claspers as stylus consisting the genitalia. Female claspers absent but cerces are present which are small and round in shape. Inside abdomen are a pair of spermatheca. So, with this, you can identify male and female species. Only females will suck the blood. It lives in warm climates, breed in rubbish heaps, cracks of walls, and soils. Nocturnal during daytime, they rest in cool, damp places. Vector for leishmaniasis is Kala Azar or visceral leishmaniasis, oriental source or cutaneous leishmaniasis, and sandfly fever. Control measures include habitat disruption, insecticides, and repellents. Next is cockroaches. Cockroaches is widely distributed pest living in close association with humans. Cockroaches harbor pathogens and allergens. Their resilience and adaptability require integrated control programs involving sanitation, exclusion, and chemical treatments. Most are uniformly black or brownish in color. Most adult cockroaches have wings which immature ones do not have. These is the image of German cockroach, Blatella germanica. So German cockroaches can develop anywhere, whether there is moderate temperature, humidity, adequate food, and water. Easily, you can see cockroaches at home if you'll not maintain hygiene or you'll keep the leftover food. Cockroaches can be easily come in kitchens. Domestic cockroaches often live in close association with bacteria, fungi, and viruses. The term domestic refers to their living in structures. Immature and adult cockroaches have essentially identical food and shelter needs. Their presence is usually indicative of poor sanitation. As I mentioned, hygiene is important to avoid cockroaches. They gain access to buildings from sewage system, lines, and manholes. Their presence is often indicative of faulty and deteriorating constructions. So cockroaches have significant adaptive features. Example, they'll survive in extended period of time without food or water and can adapt to a wide range of temperature, moisture, and light regimes which can tolerate high doses of noxious substances. Cockroaches also exhibit circadian patterns of activity where they are quiescent in light and become active in dark. They'll also aggregate in groups in cracks, crevices, and other protected places, usually near food and water. Public health importance. So, all domestic species of these cockroaches have pest status by virtue of being an offensive nuisance. Some are also associated with filth, transmission of disease, and with contact and inhalant allergies. Cockroaches are a sign of unsanitary and unhygienic conditions. Special attention should be given wherever food is prepared, served, or stored. Management and control. Domestic cockroaches, where they live near people, food, and environmentally sensitive location. So best to prevent from their gaining initial access is to these places. Preventing continuous invasion or suppressing established infestations usually involve long-term program of surveillance, pest control. Once cockroach becomes established, then insecticides alone have little long-term effect, accompanied by improved sanitation and structural upgrades. So, food scrape should be cleaned up properly. Garbage should be placed in containers with tight fitted lids, and dirty dishes should be cleaned. Openings that may harbor cockroaches should be sealed properly. It is particularly important to keep cockroaches out of commercial buildings, apartments, and other sites where they may be established. Infections and infestations come in contact with people. Next is honeybees. Honeybees are important pollinators with social colonies. Honeybee stings can cause severe allergic reactions. Management prioritizes preventing human bee conflicts while preserving bee population due to their ecological and economic values. So worldwide, there is 11 honeybee species that have been described within genus Apis. Honeybees typically live in managed hives, but escape colonies can be found in building voids, dense vegetation, hollow trees, or other location which will provide adequate shelter for them. Honeybees belonging to the order Hymenoptera and family Apidae, they are of economic importance through pollination of fruits, nuts, and vegetables, honey production, wax production, etc. This is the diagrammatic visualization of honeybee and their hives and combs. Honeybee characteristics and caste. This is the worker female. Queen is bigger in size, and this is drone, which is male. Bionomics. The stringer is a modified oviposterior, so only female honeybees can sting. Colonies will produce by swarming when the old queen and half of the workers will fly off to start a new colony. The original colony completes rearing a replacement queen. Honeybees will only fly during daylight hours when the temperature reaches to 13 degrees Celsius, when there is no rain and winds are below 19. So queen are long lived, almost around 3 years, and only one queen per colony they'll have. It will sting repeatedly because she has a smooth stinger. She is fed and groomed by retinue bees, which is their assistants, only leaves the hive for her initial nuptial flight, which is mating flight, and if she swarms with their hive, females are short lived, which lives only few weeks in summer, several months in winter, and number in tens of thousands in larger hives. So only worker bees forage for nectar and pollen. Older worker bees will forage, younger workers stay within hive and perform housekeeping chores. Will sting to defend the hive. Male are short-lived, about 6 weeks, and number from a couple of hundreds to a few thousands per hive. Their only purpose is to leave the hive, locate and mate with queens, engaging in nuptial flights. They can't feed themselves, and drones will die after mating or expelled from their hive when the onset of winter, and they are incapable of stinging in defense of themselves or the hive. Why it is of medical importance? So when the co-worker stinger is barbed and cannot be withdrawn after use, they'll encourage more bees to attack, and the stinger releases pheromones to mark a targeted individual. In hypersensitized individual, a sting can bring an anaphylactic shock and lowered blood pressures, impaired respiration, which can lead to death in minutes. So these all are the important of honeybees. Management and control efforts should be made by the general public to reduce or prohibit its access to potential nesting sites. Due to their importance of bees, it will not destroy bee swarms. It will notify beekeeper and give them an option to capture the swarm. Newly established colonies should be removed before they increase in size and become unmanageable. Bees in walls, ceilings, underfloors, down the chimneys, etc. should be removed by professional pest control operators only. Next is arachnids, which is scorpions, spiders, and ticks. Arachnids have segmented bodies, multiple legs, and adaptive venomous weapons. Scorpions can sting, causing local and systemic effects. Spiders, including black widow and recluse spiders, deliver venomous bites affecting human health. Ticks are blood-feeding parasites transmitting various diseases. Control relies on habitat management, repellent, and personal protective measures. So this Archinida is a group of consisting more than one like species. This will include spiders, scorpions, ticks, and mites. They'll adapt to kill their prey with poisons, and their bodies are divided into cephalothorax and abdomen. It will attach to cephalothorax, which are four pairs of legs and a pair of cilaria and a pair of appendages called pedipalps. These pedipalps are used for chewing and in some species for other functions. Scorpions belong to the class Archinida and are easily recognized by their characteristics lobster-like pincers and segmented tail ending in a bulbous stinger. Their body is divided into two main parts: the cephalothorax, which is fused head and thorax, and the abdominal. So, abdomen, like other arachnids, scorpions have four pairs of legs. The coloration of scorpions varies widely by species, ranging from pale yellow to dark black. They are nocturnal creatures capable of detecting predators and prey through vibrations in air on the ground. Dangerous species typically having thin, long pincers and a potent stingers that releases venom capable of causing lethal effect in their prey. Though effects on human vary, when threatened, a scorpion strikes rapidly with its tail, injecting venom into the puncture wound, while most stings cause localized pain and swelling. Some species venom may produce severe reactions including muscular spasm, respiratory failure, pulmonary edema, and shock, potentially resulting in death if untreated. So these are very dangerous species. As you can see here, this is bark scorpion. So dangerous species typically have thin, long pincers. Scorpion breeding is seasonal peaking in warm months, which also increases sting incidence. During that time, preventive measures including removing debris like rocks and firewood away from dwelling, sealing cracks and crevices around structures, and maintaining general cleanliness. Interestingly, scorpion venom contains bioactive compounds with potential medicinal uses. For example, venom from North African deathstalker scorpion is being studied for treating brain tumors and certain venom components which will show analgesic, which is pain killing properties. This is the public health significance. Management and control, as I mentioned, seal all cracks and crevices from the exterior, particularly those near ground level. Scorpions cannot climb smooth surfaces, so walls should be constructed in this fashion. Accumulation of rocks, rubbish, lumber, and firewood should be removed away from living quarters. Exposure to scorpion stings is graded during this period, so preventive measures should be initiated before or during that time. Breeding is seasonal and generally occurs during the warm months. Potential benefits, as I mentioned, North African deathstalker scorpion has shown potential in treating brain tumors. Some venom also has pain killing potential, example, black mamba venom. Next is spiders. Spiders are another prominent group with Arachnida with about 35,000 named species globally. They are primarily nocturnal and generally avoid humans, biting only accidentally when disturbed. Spiders have a two-part body structures, the cephalothorax and abdomen, connected by a narrow pedicel. They have four pairs of legs which lack wings and possesses Chelicera, which is basal segments of which contain poison glands used to subdue prey. The venom paralyzes or kills prey, which is then digested externally by injecting digestive enzymes. So, only a small number of spider species are considered dangerous to humans, notably the black widow and the brown recluse. Black widow spiders have shiny black bodies with a distinctive red hourglass marking. Their venom is neurotoxic, causing severe muscle cramps, pain, headache, nausea, and sometimes raised temperature. Brown recluse spiders cause skin necrosis due to dermonoractic venom. So, spider bites ranges from minor localized regions to severe systemic illness depending on bite location, venom quantity, and individual health. Spiders tend to live where their prey and shelter are available. They are mostly outdoor dwellers but can appear indoors seasonally. Related to mating, control is approached by maintaining cleanliness and managing other insect populations to reduce spider food sources. So morphology, as I mentioned, spider belong to the class Arachnida. Order are Araneae. All are wingless and have four pairs of legs. Their body is divided into two main regions, cephalothorax and abdomen, which is joined by a narrow connection, the pedicel. Poisonous glands are usually located in the basal segment, and the captured prey is usually paralyzed by the venom or crushed with the strong chelaceae, and digestive fluids are introduced via the maxillary glands. Public health significance, almost all spider bites cases occur when humans accidentally come in contact with them. They'll not attack people as in popularly visualized. Effects of the spider bite range from benign to severe depending upon complexity of factors such as site of bite, amount of venom injected, age, and physical health of the person. This is the black widow spiders. Adult females are black and shiny with a red hourglass marking on the underside of the pea-sized abdomen. The males are usually black and much smaller with brightly colored chevron-like bands and spots on the abdomen. Most dangerous species are found in every terrestrial and freshwater. It will feed on fungi, plants, and animals. Its venom is neurotoxic, affecting the CNS. Cramps in the legs, arms, and chest occurs together with local swelling. Other symptoms include headache, nausea, tremors, and slight rise in temperature. This is the public health significance. Management and control. Spiders usually reside where arthropod prey and in adequate shelters are available. Most prefer outdoors, but the periodic appearance or presence of wandering species, spiders include inside dwellings, is seasonal and usually related to their mating habits. Frequent house cleaning and control of other insect pests often will limit their numbers. Next is ticks. Ticks are blood-feeding ectoparasite found on reptiles, birds, mammals, and occasionally amphibians. They belong to two main groups, hard-boiled and soft-boiled, distinguished by the presence or absence of a dorsal scutum. Hard ticks have the capitulum located anteriorly, while soft ticks' capitulum is positioned ventrally underneath. So ticks have 6 legs in the larval stage and 8 legs as nymphs and adults. Most hard ticks undergo a three-host life cycles where the larva, nymph, and adult stages each feed on potentially different vertebrate hosts. Ticks have significant public health importance. This is the western black leg ticks. So ticks have significant public health importance because their bites can cause skin conditions, autoascariasis, tick paralysis, and allergic or anaphylactic reactions in sensitized individuals. Crucially, ticks transmit a variety of bacterial, viral, and protozoan diseases such as Lyme disease, Rocky Mountain spotted fever, tick-borne encephalitis, and babesiosis. Control, this is the ticks disease which will cause in human. Control and management of ticks involve natural regulation by climate and predators, chemical control using acar acaricides, personal protective measures like repellents and protective clothing, and environmental modification to reduce tick habitats. Tick removal must be done carefully using tweezers to grasp the tick close to the skin and pull steadily outward to minimize pathogen transmission. Early removal reduces infection. Personal protective measures including avoiding tick-infested areas, wearing light-colored long clothes, tucking shirts and pants appropriately, applying DEET containing repellents to exposed skin, and treating clothing with based repellents. So these are the control and management: natural control, chemical control, avoidance of ticks, personal protective measures. Summary for lab diagnosis of parasites. So parasites are organisms that derive nutrition from a host without compensation. Laboratory diagnosis is crucial for confirming suspected parasitic infections and detecting asymptomatic cases. Specimens for diagnosis including stool, blood, serum, and other body fluids or tissues. Methods used include microscopy, which is staining and concentration, culture, molecular techniques like PCR, restriction fragment length polymorphism, and serological assays. Stool examination detect helminthes, eggs, protozoan cyst, oocyst, blood smear detect malaria parasites and other bloodborne parasites. Malaria diagnosis involving preparing thick and thin blood smears, staining GIMSA, and microscopic examination. Thick smears allow detection of parasites by lysing red blood cells but can distort morphology. Thin smears are fixed and allow species identification and parasitemia quantification. Laboratory diagnosis involve treatment and public health surveillance but depends on skills, equipment, and quality of specimens. So this is the summary for laboratory diagnosis of parasite, and this summary is for medical importance of arthropods. So arthropods are invertebrates with jointed legs, segmented bodies, and chitinous exoskeletons. Anatomology studies insects' roles related to humans, including disease transmission. Medically important arthropods transmitted disease via different mechanism, which is biting, mechanical transport, vector-borne transmission. The success arises from small size, rapid reproduction, diverse habitats, and evolutionary adaptations. Main habitats including soil, water, air, which is temporary, human, ectoparasites, animals, and plants. Control measures include personal protection, environmental manipulation, biological control, chemical control, and legislative measures. Modes of disease transmission include direct contact, mechanical transport, and biological transmission, which is propagative, cyclopropagative. Vectorial capacity depends on abundance, host preference, reproduction, longevity, dispersal, and competence. Important arthropods include mosquitoes, flies and fleas, sandflies, cockroaches, bees, and arachnids, which is scorpions, spiders, and ticks. Diseases transmitted include malaria, dengue, filariasis, leishmaniasis, plague, typhus, and various viral infections. Control measures will focus on vector eradication, habitat removal, repellents, insecticides, and personal protective behaviors. Question part for 3 mark. Definitive define parasite and host. Second, list 4 types of specimens used for parasitic diagnosis. Third, explain the difference between thick and thin blood smears. Fourth, what is the purpose of the Giemsa stain in malaria diagnosis? Fifth, name 2 molecular techniques used for parasitic infections. Sixth, what are the main categories of medical important arthropods? Seventh, list 3 habitats where arthropods can be found easily. Eighth, define vector and give 2 examples of each. Ninth, state 2 advantages and disadvantages of microscopy in parasitic diagnosis. And last is 10th, what are the characteristics of insect that distinguish them from other arthropods? Questions for 5 marks. First, describe the life cycle stages of mosquito and their relevance to disease transmission. Second, explain how visceral and cutaneous leishmaniasis are transmitted. Third, discuss the principles and steps involved in preparing and examining a malaria blood smear. Fourth, outline the modes of disease transmission by arthropods and give example. Fifth, describe the impact of vectorial capacity on disease transmission and list factors that influence it. Sixth, explain the biology and medical importance of ticks, including their control methods. Seventh, what are the structural and functional adaptations of arthropods that contribute to their success? Eight, discuss the roles of flies as mechanical vectors in human diseases and methods to control them. Ninth, explain the characteristics and medical importance of cockroaches. And last is describe the life cycle habitats and controls of sandflies and their roles in disease transmissions. So with this, we have covered fully parasitology part, everything in that. Last topic is also covered, which is laboratory diagnosis of parasitic infection and arthropods. So today's class will wind up with this parasitology part. Thank you.